How can a molecule only 21 to 24 nucleotides long regulate multiple genes? Well, the answer is by guiding a silencing complex to messenger RNA and reducing the production of proteins. This tiny regulator is called microRNA or miRNA. It's a small, non-coding RNA molecule that regulates gene expression without producing a protein itself. When microRNA genes are encoded, they are transcribed into a longer molecule that is called primary microRNA or pri-miRNA, which falls into a hairpin structure inside the nucleus. The pri-miRNA is processed into a pre-miRNA. It's then transported to the cytoplasm where further processing produces the mature microRNA. The mature miRNA associates with the Argonaute protein and becomes part of a silencing complex called RISC. The microRNA guides the RISC to a specific mRNA by recognizing a complementary sequence. Once they bind, the target mRNA may be cleaved and degraded, or its translation may be inhibited. In both cases, less protein is produced. This is called a post-transcriptional gene regulation because it occurs after the gene has been transcribed into mRNA. In plants, miRNAs often bind their targets with high complementarity, so direct mRNA cleavage is common. While in animals, pairing is usually more partial, making translation repression and mRNA destabilization more common. But why miRNA is especially important in plants? That's because some of their targets encode transcription factors. If environmental stress changes the level of a particular miRNA, the transcription factor it targets may also change. And because transcription factors regulate many downstream genes, this can influence a broader gene regulatory network. This becomes especially important during abiotic stress such as drought, salinity, or heat. One important pathway can be summarized as one microRNA affects one transcription factor that affects many downstream genes. These downstream genes may be involved in hormone signaling, ion homeostasis, root development, antioxidant defense, or other stress responses. However, an increase in miRNA does not automatically mean greater stress tolerance. Its effect depends on the plant species, tissue, developmental stage, stress conditions, and specific target genes. So the key idea is simple. A microRNA may be only 21 to 24 nucleotides long, but it can influence multiple genes and help reshape how a plant responds to its environment.